Welcome to the tutorial session. So today's session we are going to look at some problems which are discussing concepts on drug receptor interactions, and also we will look at some stereochemical issues, either conformation of cyclic and acyclic structures. So to begin with, let us look at some drug receptor interactions. So the first question is, draw out a dose response curve for a full agonist. So we have already studied, or we have already looked at, that agonist is basically a compound that binds to the receptor, right, and it also generates a signal, much like the natural ligand does. So an agonist is essentially close to or very similar to the natural ligand. So here is a dose response curve for the natural ligand. And so what we are looking at on the y axis is the percent muscle contraction, and the x axis is the concentration. So we have already seen this in class, and so the dissociation constant Kd is shown here, which is basically 10 power minus 8, right, molar. Now the question here is, draw out the dose response curve for a full agonist. Okay. So here what we mean by the word full agonist is that it binds, or it carries out the function identical to the natural ligand. So a full agonist would be essentially the same as the natural ligand. Okay. And so if the percent muscle contraction is elicited by binding of the natural ligand, then for the full agonist we would have an identical curve. Okay. So the dose response curve for a full agonist would be essentially identical to that of the natural ligand. The next question is, estradiol and tamoxifen. Estradiol, which is shown here, is a carbon rich steroid, okay, and tamoxifen, which is a clinically used drug, mimics the shape and chemical composition of estradiol. Okay. So you see that there are some similarities in the functional groups, and it acts as an antagonist. So tamoxifen is actually an antagonist. Let us write that down, antagonist, okay, to estradiol. Now the metabolite of tamoxifen, that is shown here, is an agonist, right. So let us recall what an antagonist and an agonist is. Antagonist inhibits the function of the receptor, or reduces the signal transmission by the receptor by binding to the same site pretty much as the natural ligand. An agonist, on the other hand, competes for the same site, but it increases or it elicits the same response. So the difference between the antagonist and agonist in this case is that one has a function which is exactly opposite to the other. So the question here is, what is your inference from this? So to recap, we are dealing with a commercially or clinically used drug, tamoxifen, which is an antagonist, and it inhibits the binding of estradiol, and its metabolite, which is shown here, which acts as a agonist. So the inference from this is that there must be some binding group which is obviously binds to estradiol, which binds similar over here, right, and whatever the binding that happens with tamoxifen does not induce the conformational change required for the signal to be transmitted. Okay. So in the case of tamoxifen, it binds to the receptor binding site, but it does not induce the conformation change that is required for the signal to be transmitted. But the metabolite of tamoxifen binds to the same site and induces the same conformational change that estradiol perhaps does, and is able to act as a agonist. So this is the major inference from this data. To look at it somewhat at a molecular level, let us look at this picture over here. It is known that these residues, aspartate, glutamate and arginine, are important in binding to the natural substrate, And so, what happens in the case of tamoxifen is that this dimethyl amino functional group, which is protonated, binds to or weakly interacts with aspartate, and the rest of the molecule mimics the shape and size of estradiol. Whereas, the metabolite, as you can see here, has an extra hydroxyl group over here, and this interaction is gone. So, this extra hydroxyl group makes it much closer in structure to estradiol, and perhaps this mimics the natural way of binding, and it starts becoming an agonist. Okay. So, this is an example of how making of a compound which can act as a drug on its own, but once it gets metabolized, some part of it can actually function very differently, in fact opposite to what you would want it to work on. So, therefore, understanding how drug undergoes metabolism and what the byproducts are is an important and crucial part of drug discovery. In the next question, we will look at some stereochemistry. What is the relationship between this compound here and this compound here? Okay. So, in order to do this, let us first determine the absolute stereochemistry. So, the absolute stereochemistry of this molecule, we need to assign the priorities. So, this would be priority number 1, this will be priority number 2, this will be priority number 3. So, hydrogen is behind us. So, this would be R. Okay. So, priority for this molecule similarly, we can assign it based on OH is 1, COCH3 is 2, ethyl is 3, and so we need to look at it from here. If you look at it from here, 1, 2, 3 clockwise. So, this would also be R. Okay. So, these two molecules are identical, they both have R configuration. So, in the next question, we will go back to our receptor binding, ligand binding. So, when a new drug candidate is tested, the following profile was observed, where you have percentage muscle contraction to be 35%. Right. So, what is your conclusion from this? So, in order to address this question, we would assume that the natural ligand here, which is acetylcholine, has 100% muscle contraction versus concentration. And so, if this is the profile of the natural substrate, the profile of the unnatural substrate looks something like this, right. So, if this is 50%, then it is less than 50%, and so this would be a partial agonist. Okay. So, the partial agonist has both the properties of an agonist as well as an antagonist. Okay. So, if it is a full agonist, then we have already looked at that whatever conformational change that occurs with the natural ligand will also occur with the full agonist, and you will see a nice increase in the muscle contraction. But here, since it is acting both as an agonist as well as an antagonist, you do not see complete muscle contraction in the dose response curve. So, therefore, you call it a partial agonist. Okay. So, the next question is propranolol is an adrenergic antagonist. Compare the structure of propranolol with noradrenaline and identify which features are similar in both molecules. Okay. Then suggest why this molecule might act as an antagonist rather than as an agonist, and whether it might show any selectivity between the different types of adrenergic receptors. So, in order to address this question, first let us redraw propranolol, and if we redraw it and in a conformation that is very similar to noradrenaline, we find that there are obviously there are a number of similarities in this. So, as shown here in red, this is the, so here is the active structure as shown here, and what we have in addition is this extremely bulky naphthyl group, which is going to be quite hydrophobic, and there you do not have that kind of a group, and you have this two hydroxyl groups which are going to have hydrogen bonding interaction perhaps. Okay. So, therefore, it is possible for this moiety in both molecules to form similar interactions with the receptor. Okay. but because the aromatic systems are very different and they are so different that it is likely that it will act as an antagonist rather as an agonist. Okay. Because it is quite unlikely that the receptor will have the same induced fit from binding to noradrenaline or as well as propranolol. So, it is quite possible that this compound will act as an antagonist. Now, in order to understand whether it is going to have selectivity, we would need to find out whether this molecule, how this molecule varies across various isoforms of this receptor, and that it may not be easy to predict beforehand whether it would have selectivity or not. Next question is, what is the relationship between these pairs of compounds? Okay. So, most of you are familiar with the Newman and the Sawhorse projections. So, this is the example of a Sawhorse projection. So, we have numbered the carbons in the following way 1, 2, 3 and 4. So, now in order to address this question, first let us look at the first molecule, and if you see the carbon number 1 over here, the priorities are shown as shown here 1, 2, 3, and so this would be clockwise and therefore, it would be R. And if you take this other carbon, if you assign the priorities, Br is 1, the second carbon is 2 and this methyl is 3, and so you have anti-clockwise, but since hydrogen is on in the opposite side, we would call this as R. Okay. In the second example, we have looked at the first carbon over here, the stereochemistry over here would be we need to be determined. So, in order to determine this, we will look through the thing. The priorities first is 1, 2 and 3, and so now we can look at it from one angle, and it looks so if you look at it from the opposite side of the hydrogen, then you have 1, 2, 3, this is anti-clockwise, and so it would be S. Right. And similarly, if you look at the other carbon over here, this the absolute stereochemistry is 1, 2 and 3, and so look at it from here, then the priority would be 1, 2 and 3. So, this would be clockwise, and so it would be R. So, to look at the stereochemistry, we have 2 R and 3 R, whereas in the other case it is 2 S and 3 R, and so these are diastereomers. Okay. So, some of these questions you will have to practice on your own to get better at it, but we should be able to assign the absolute stereochemistry with comfort, and only then we will be able to determine whether they are identical or diastereomers. Next question is, draw out a dose response curve for a competitive antagonist in the presence of a fixed concentration of the natural ligand. And here the fixed concentration of the natural ligand will give us elicit maximal response. Okay. So, here is the dose response curve that you would see for a competitive antagonist. So, you see the percentage muscle contraction at a very high ligand concentration, you will see 100%. So, when there is 0 or no competitive antagonist present, you do not see any difference in the or you see 100% dose response. And as you increase the concentration of the competitive antagonist, you will find a dose dependent decrease in the muscle contraction. And so, then we can determine what is known as IC50, which is basically inhibitory concentration 50%, which is the concentration required to inhibit the activity, or in this case muscle contraction, by 50%. So, this is how the dose response curve for a competitive antagonist in the presence of a fixed concentration of the natural ligand will look like. So, the next question is, draw out a dose response curve for a competitive antagonist in the presence of the natural ligand. That means that it is not a fixed concentration of the natural ligand, but you vary the concentration of the natural ligand and keep the competitive antagonist at a fixed concentration, but you can vary it. For a particular experiment, you will have a fixed concentration, for the next experiment, you will have the higher concentration. So, this is how it looks like. Again, we have seen this in class. So, in the absence of any antagonist, this is how the dose response curve would look like. And as you add a competitive antagonist, then the curve moves towards the right, which means that the let us say this is the 50% thing, it keeps increasing, and keeps moving towards the right. Okay. So, this is much like a competitive inhibitor that we have seen in enzyme inhibition, and where the dose of the ligand goes up, at some point you will see maximal activity. So, this is how a competitive antagonist will look like. In the previous case, where we saw in the partial agonist, we saw that even if you keep adding the ligand or the drug, you never see a situation where you see 100% muscle contraction. Whereas here, if you keep adding large excess of the natural ligand, you get complete activity at high doses. So, the next question is, what is the relationship between cis 3 methyl cyclohexanol and trans 3 methyl cyclohexanol? So, they are non superimposable, I mean they are not mirror images, and so they do not have a mirror image relationship, but they differ in stereochemistry, and therefore, they are diastereomers. So, the next question is, what is the relationship between this compound here and this compound here? So, again I like to follow the systematic method of doing this. So, we will first assign the stereochemistry of the carbon ahead. So, that turns out to be S. We can go through the process. Now, we do the same thing for the back carbon, and you get the stereochemistry of that to be R. Okay. And this is for the left hand side molecule. Now, for the right hand side molecule, we can assign the stereochemistry for the front carbon, which turns out to be R, and the back carbon, that turns out to be S. So, what we are dealing with here is that this carbon is S, which is exactly the opposite, and this carbon is R, which is exactly the opposite, and so we are dealing with enantiomers. Okay. So, another way to do this is to write the structure in the Newman projection and compare it. So, if you see draw the Newman projection for the molecule on the right, you get the following conformer, and then you can actually draw a mirror plane, and you will find that these two are exactly mirror images, and therefore, you can conclude that they are enantiomers. The next question is, a compound has been identified to be a non-competitive antagonist. Describe how the dose response curve for this compound in the presence of the natural ligand will look like. So, again we have looked at this in class. So, for a competitive antagonist, you will find that at very high concentrations of the natural ligand, you will find that there would be 100% activity. So, for a non-competitive antagonist, the maximal response will go down. Okay. And this happens, as you can see here in this plot, as you increase the dose of the ligand, the response goes down. Okay. So, this happens because it is possible that there are two different binding sites may be involved. So, when the non-competitive antagonist binds to, let us say, an allosteric binding site, a site to which the endogenous ligand normally does not bind to, what may happen is that this might induce a conformational change. So, let us say your receptor is binding to this site over here. So, this is your receptor binding site, whereas if your inhibitor binds over here, then what it can do is it can induce a conformational change here, which will prevent the ligand from binding. Okay. So, but this does not happen to a great efficiency, and therefore, you will find that there is still some activity, that is residual activity, that happens even if you increase doses of the compound. So, the way in which we distinguish a non-competitive and a competitive is that when the ligand is competing for the same site, then when you increase the ligand concentration, or even increase the natural ligand concentration, at some point it will displace all the drug, and it should be able to restore the activity, or the activity of the receptor should be 100%, or the phenotype that we are looking at here is percentage muscle contraction should be 100%. But when we are looking at a non-competitive situation, then since it does not bind to the actual binding site, and it does something else which induces a conformational change in the protein, which affects the binding of the endogenous molecule, what ends up happening is that you will see a reduction in the activity as you increase the dose of the non-competitive inhibitor.